Hello.
Hey, how you doing?
Good. How are you?
Pretty good, pretty good. Thank you. Sorry about that. I had to get my questions all sorted out and stuff. All
It's all right.
right, cool. Well, you can start first if you want. Ladies first.
Okay. I think we still have to introduce ourselves. So I'm Zaina.
Oh yeah, I'm Nick Parham. Um, we have two. Oh no, Yvonne dropped the class. The
Yeah.
other female in the group, so she's not here anymore. And I don't know what happened to the other guy. I forgot his name.
Um. I think it could just be because everything with the at least with this being due so soon, we just he
Yeah,
just wasn't able to read the messages.
yeah, because I honestly thought that it was gonna be due like on the weekend again, but
Yeah. Honestly, I didn't even know we were doing one this unit because last unit, it had the due dates kind of like ahead of time.
yeah, exactly, yeah. But this one kind of snuck up on us, yeah.
Yeah.
But yeah, you can start though if you want.
All right, let's see. What are the steps of meiosis?
The steps of meiosis.
Yes.
Um, the stages of meiosis. So you're not talking about mitosis, right?
No, no, the other one,
The other one.
the
Hmm,
longer one.
the longer one. Like you just want the names of the different stages, right?
Yeah.
Okay. Let's see. Mm, give me a second. I'm looking it up on my
Yep,
notes
take your
right
time.
now. Meiosis. Okay. Stages are um, prophase,
Hmm.
anaphase 1, anaphase 2, metaphase 2, metaphase 1, telophase 1, telophase 2, meiosis 2, anaphase, interphase, metaphase, telophase 2 and psychokinesis, telophase 1 and cytokinesis, and prometaphase 1.
So I have it written as because I think it's more as like prophase one, metaphase one, anaphase one, telophase one, and then that's where cytokinesis comes in. And then it goes to prophase two, metaphase two, anaphase two, telophase two, and then cytokinesis again.
Yeah, wow. Yeah, so that one's a bit confusing.
I kind of, the way I just remembered it is kind of like with mitosis. Mitosis has the exact same steps, the prophase, metaphase, anaphase, and telophase. It's just meiosis repeats them again.
Oh, wow. That's interesting. Man,
Yeah.
that's crazy. Okay. So my first one is, do prokaryotic cells have a nucleus?
Prokaryotic cells. Give me 1 second. Okay. No, they don't have a nucleus.
Correct. Yes, they do not have a nucleus.
Okay. All right. And let's see. And then what's another name for S phase?
For S phase,
Yes.
and is that part of mitosis or meiosis?
So it's basically, like it, um it's basically about the cell's life, not exactly mitosis and meiosis. So it's like what the cell is doing 90% of the time.
Oh, so cell function.
So technically, the other name for S phase is interphase. So that's basic,
Interphase.
yeah. So that's basically when the cell is doing most of its life growth DNA replication and carrying out its daily cell functions.
Huh. Oh, another word for it can also be interkinesis, right?
I think so. I didn't write that down, but I think so.
Okay. Yeah, because that's what I got, interkinesis.
I think it came in later on towards enzymes when they started mentioning that S phase could also, like interphase, could also be known as S phase.
Can also be known as S phase. Okay.
Mm-hmm.
Cool. Okay, but is interkinesis, it could also be technically used, or
That I don't know. That would be something to make a note of, and we'll just look at that later.
okay. All right. This is my second question. Is bacteria a unicellular or multicellular prokaryote?
Ooh, I think it's unicellular?
Yeah,
Cellular? Okay,
correct. Yeah, all bacteria is unicellular prokaryote cells.
got it.
Prokaryote cells.
Um, let's see. I had it. I just gotta find it again. Oh, okay. What's the difference between lactose and lactase?
Lactose is when like let's say I'm allergic to milk.
Mm-hmm.
They say lactose intolerant. Lactose is I want to say lactose is the cells that are found in milk, or like uh how can I describe this? Basically like a group of cells. I would say lactose is like a group of cells within like certain products like milk and stuff.
Mm-hmm.
And when people say lactose intolerant, I think that like has something to do with the translation and transcription of the lactose and the lactase. I forgot the definition of lactase, but yeah, lactose, I would think it would have something to do with the cells that are inside the milk. And then if you're lactose intolerant, like your genetic makeup may not be compatible with the lactose.
Mm-hmm.
If that makes sense.
So the kind of like how what it would be, it's essentially is that lactase is the enzyme that you need to break down lactose. So
Oh,
without that enzyme, lactase, then you're lactose intolerant.
so lactase is needed to break down lactose.
Yeah.
Okay, cool. And without lactase, you are lactose intolerant.
Mm-hmm.
Lactase. Wait, lactose. Okay.
Yeah. So
Lactase,
lactase
lactose.
was, so I guess the trick they kind of learned, like with enzyme, it's an enzyme because it has ASE after it.
ASE after it.
Yeah.
Okay. And lactose has OSE. Okay.
Mm-hmm.
Cool. Thank you. Third question is, why is gene expression regulated?
Gene expression. Let's see. So that one, I don't know.
Okay. Basically, gene expression is regulated so that specialized cells only produce the proteins necessary for a specific function.
Okay.
Yeah, it's a very long, drawn out definition, but
Got it. That makes sense
yeah.
because you're
So
trying
only
to.
yeah, so only certain cells are going like certain parts. So
Got
only
it.
like certain cells produce the proteins necessary for like a specific function within the body. Like
Okay.
maybe
To
like
keep everything regulated.
yeah, pretty much, yeah. Like so like the movement of the arms and legs, I think are
Mm-hmm.
separated and differentiated by specialized cells, like
Okay.
and the proteins that are in the cells. Yeah,
Got it.
yeah, it's pretty cool.
All right. Let's see. What is the What is the difference between genotype and phenotype?
Cool. Hey, this is surprisingly that was actually one of my questions too. So that's awesome. Yeah, a genotype is the genetic makeup inherited by an organism, whereas a phenotype is the expression of the gene.
Okay. Yep. Perfect.
Cool. What is an enzyme?
An enzyme is basically it's a protein that acts as a catalyst in chemical reactions. So it can like speed up like rates of chemical reactions like photosynthesis, protein synthesis, and it remains unchanged at the end of the process.
Huh. Okay, yeah, it's kind of like resembles what I put for the answer, which I put a group of proteins that drive reactions inside cells, making life
Okay.
possible. Yeah, so
Yeah.
it's pretty much the same general gist. Yeah, that's cool. Okay, cool.
What are some examples of enzymes?
Um, hmm, like amino acids. Like, would that be a form?
So amino acids wouldn't be an example, but what would break it down to amino acids? Amino acid is like that end result. It's not the enzyme.
Oh, would it be protein synthesis?
Yeah, it would be the protein. Yeah.
Okay. So protein synthesis it essentially breaks down the amino acids and different proteins inside the enzyme.
So essentially it would be like so I think that so the actual term would be like protease, and so it's not protein synthesis. Yeah, maybe it's around kind of similar to that. I would have to look back to protein synthesis. But essentially the actual name for the enzyme is protease.
Protein.
So it breaks down like the proteins to amino acids, which if we thought back to the first unit, the amino acids were proteins monomers. So
Yeah,
that was very interesting.
that is interesting. Yeah. Huh.
And then like another thing, if you were to think back to like the biomolecules, you have like the protein carbohydrate, carbohydrates. So again with the ASE, which is breaking down carbohydrates to glucose. And then lipase, which is like, back to lipids and everything, but the lipase would break the fats down to the fatty acids and the glycerols.
Huh. Okay. Cool. Thank you. Did not know that. When can a chromatin be ready by transcription factors which are critical for gene expression? Yeah, that was some
Transcription, that was actually a pretty
cool one, right? Yeah.
cool, but it was hard for me to fully understand what it was.
The concept of it, yeah.
Yeah. Let's see.
Well, I look at it as translate is to like express something in a different way. So I would think like transcript is like a transcription, like transcripts for high school. It's like a records, like a record, like to keep a record of all our high school classes. Like I think it's a record. The transcription is a record of all the proteins inside inside the cells that are being active and interacting with each other during gene expression.
Okay.
Does that make sense? Yeah.
Yeah.
It's a very difficult concept, but yeah.
That was a lot.
Yeah,
and then your question was like in relation to the chromatin.
Wait, what did you get?
Oh no, just go with like your question you were asking.
Oh yeah. Oh, you were providing the answer.
No, I just can you re-ask the question?
Oh, ask the question.
Yeah.
When can a chromatin be read by transcription factors which are critical for gene expression?
When can they be read? Is it when the ribosomes come in? Because like the ribosomes read them?
No.
So then that one I don't know.
It's when the chromatin is not super coiled.
Okay. So when it's stretched out.
Yeah. Yeah, that one was a little tricky one.
I'll write that down. Okay. All right. Let's see. So what is the main purpose of when cells multiply? Like, why are they trying to multiply?
I think that cells are trying to multiply, are always trying to multiply because, let's say a cell gets broke, like let's say some type of damage happens to a cell or a cell
Mm-hmm.
dies, it replicates itself so that it can continue to go on even if the original cell died off. It still
Yeah.
has copies of the original, so you
Mm-hmm.
keep going in a way. If that makes sense.
Yeah. Keep everything going. Yes.
Yeah. Um, what are the first two things that must happen before gene expression can take place? Sorry about that. I got a little butter in my finger.
No, it's all right. The first two things that have to happen before gene expression takes place. Mm, that one you're gonna have to let me know. I don't know that one.
The first two things that must happen before gene expression can take place are one, transcription, and two, translation.
Ah, got it.
Okay, cool. All right, it's your turn. You can ask a question now.
Let's see. What exactly is the central dogma?
Ooh, the central dogma. Um, the central dogma. Here, let me just look it up in my notes right now. Central dogma. Central dogma is essentially DNA that is converted into a functional product.
Okay. So I kind of have it written as kind of a little more extensive. Basically, like DNA makes copies of itself by the process of replication. And then
Wait, can you repeat that last definition?
DNA makes copies of itself by the process of replication. Then they pass the code, then the code of DNA is cracked and transcribed into a messenger of RNA. And then mRNA is the reads the code and translates it into protein. So essentially, it's basically just replication, transcription, and translation all in one. Like
All at
you're adding
once?
all, not all at once, but all in one. Like you're fully describing the process of all three of them.
Oh, okay,
Yeah,
okay.
it's all three of those processes.
In one term. Huh.
Yeah.
Okay, cool. All right, got one more question. What is the fluid mosaic model?
Fluid mosaic. I remember that, but let's see if I wrote it down. Because I remember it being talked about, but which module was that one in? If you know.
I'm pretty sure it's the enzyme one.
Is that where
Would that be called it would be called transcription, translation, and gene expression, right?
yeah. Oh, but the fluid mosaic, I want to say it has something to do with how it's always moving, like the um I don't know, I feel like it has to do with how the cell is like always moving or the process, like it's very fluid type of thing. That's what I think it is.
Yeah, it has somewhat to do with movement. What I got was that it's a phenomenon in which the cell membrane is flexible to where proteins can move around, but yet it stays intact to maintain a consistent barrier.
Mm-hmm.
Cool, cool. That is pretty cool though. How that's able to
Yeah.
do that. So it means that like the cell membrane is really strong then.
Uh-huh. That I like
Okay.
when I heard that. I remember it was in one of our readings we had to do, and it brought up the fluid mosaic. I'm pretty sure that's where it was.
Yeah.
I just can't find it in the notes. But what you said, I think that sounds exactly what it said.
Yeah. Hey, you got one more question?
Um, let's see. Why are temperature and pH important to enzymes?
Mm. I believe that temperature and pH are important to enzymes because different cells have different regulatory techniques for keeping the temperature balanced and it was temperature and which one?
pH.
pH. pH because the enzymes essentially like the cells have water in them, I would think, right? Because pH has to do with like water levels and stuff. So I would think that it's important to the enzymes because it helps regulate the heat and the water levels for some cells inside the enzymes.
Yeah, so essentially it would be like enzymes are very, it says very sensitive, but I would say like more enzymes are very picky because each enzyme needs that perfect temperature and perfect pH to perform the function that they're trying to do. And if they don't have that, then they can't perform those functions.
Okay, each enzyme needs specific temperature. It was temperature and what was one again? I'm sorry.
pH.
pH
Hmm.
to properly
To
do
be
the
able
job that's supposed to
to, yeah, to be able to do their job.
really be able to do their job. Hmm. Okay. Cool. Thank you so much, Zaina.
You're
It was
welcome.
cool. Yeah, it was awesome. This was a great session.
Yeah, and thank you.
Yeah,
Definitely
cool.
I wrote some stuff down that I'm like, I need to look over again.
Yeah, me too. I'm about to go look that up tonight when I get off work. Yeah, all the stuff
Hmm.
that you helped me get reclarified on. Thank you.
You're welcome.
But for sure, hopefully, I forgot his name, but hopefully he can be here for the next session next month.
Yeah. Well,
I'll
we have the
I'll
dates
message him
now.
for sure.
Yeah, and we can plan ahead that time to make sure everything.
Is in order, yeah, because yeah, we
Hmm.
kind of had to just hop on this on the fly. Yeah, I'm sorry about that.
Yeah. Oh no, it's okay. I did too. When you said you could only meet at 1 at first, I was like, man, I don't I work till 2.
Yeah, and I was like, I work at 4, so like I was glad I was able to do this before I went to work. Thank you.
Yeah.
All right, cool. So I'll see you next month. All right,
See
take
you
care.
next month.
Bye bye.
Bye.